everyone. This is Dr. Man from Pathology. Shall we proceed to... Yeah, okay, never mind. My screen video is very faint, but all right. I'm going to introduce my next topic, which is on RNAscope. All right. This is a very interesting new technology. So the title of the talk will be Introduction to RNAscope Technology, and just a short synopsis. This is a novel multiplex nucleic acid in situ hybridization technology, and it represents a major advancement in RNA-ish approaches in terms of investigation, with a special probe design that can amplify target-specific signals and suppress background noise. It's been proven in over 2,800 publications and has been utilized in multitude research areas. So it's with great pleasure I introduce Dr. Amrita Roy, who is the Product Manager of Advanced Cell Diagnostic Portfolio at Biotechne. Dr. Roy is very experienced. She obtained her PhD in protein science and molecular biology from National University of Singapore and has a postdoctoral experience of 5 years. She has published in many peer-reviewed journals and has vast experience in nucleic acid in situ hybridization technology. So, Dr. Amrita, the floor is yours.
Thank you very much, Prof. Man. Thank you for providing me the opportunity to introduce this technology to the group. For bandwidth issue, I'll now switch off my video while I'm presenting, and then I'll come back live after the presentation. Hope that's fine. Title says, I'm going to talk to you about the next generation in situ hybridization technology, which is RNAscope. I'll give you an overview, the clinical and research applications. So to begin our discussion, we will start with the traditional in situ hybridization. In situ hybridization is very much a complementary technique to immunohistochemistry or IHC, but rather than using antibodies to detect proteins like you do in IHC, in situ hybridization leverages on probes to detect either DNA or RNA targets. And this is done through hybridization of the probes to their complementary sequences. DNA-ish is typically used to assess the chromosomal aberrations like translocations, fusions, or different kinds. One classical example is involving the translocation of chromosome 9 and 11 that lead to BCR-ABL gene fusion. And another application could be to assess the copy number. Perhaps the most common example is the HER2 in situ hybridization assay, where HER2 can be amplified in breast cancer as well as in other cancers. On the other hand, RNA in situ hybridization uses shorter probes that target typically the RNA and used to assess gene expression, which is closely correlated for protein expression, and protein expression could be measured using immunohistochemistry. So now, as we already have immunohistochemistry, the question arises, why would someone choose to use RNA in situ hybridization? There are a number of instances where it is advantageous to look at the RNA versus the protein. One such classical instance is when protein don't lend themselves to the creation of good antibodies. An example of that could be the E6, E7 oncoprotein of human papillomavirus or HPV. Both these oncoproteins don't lend themselves to create good antibodies. And therefore, in this case, RNA in situ hybridization is a great alternative to the P16 immunohistochemistry, which is a surrogate marker to HPV involvement. Another example is where RNA don't encode protein themselves, such as non-coding RNA, circular RNA, microRNA, and therefore there is no available antibodies to detect these kind of targets. Nonetheless, RNA can also serve as a biomarker for given process like infectious disease, something like to detect the emerging pathogens, like what happened in COVID-19. For SARS-CoV-2, when the pathogen came, the pandemic occurred, we didn't have any antibodies to detect that. We could quickly go back and generate RNA probes for that and that pathogen, and that RNA probes could be used to generate vaccines, like what we have done for Moderna to develop their RNA-based vaccine. All right. So since its introduction in 1916, in situ hybridization has undergone significant advancements. Initially, in situ hybridization was developed using radioactive copies of DNA sequences to detect complementary DNA sequence. However, the challenge with this technology was that it posed safety hazards, required prolonged exposure times, and had limited resolution. As time went on, advancements in technology allowed the shift to non-radioactive methods like in situ hybridization with the fluorescent version, which is called fluorescent in situ hybridization or FISH, which improved the safety and increased the sensitivity as well. Now, in 1980s, RNA in situ hybridization was identified, and it enabled us to be able to both locate and understand the expression of RNA within the spatial context of tissues. Fluorescent in situ hybridization or FISH also gave way partially to newer techniques, namely silver or chromogenic in situ hybridization and RNA in situ hybridization. Because of all this advancement over the years, ISH or in situ hybridization became widely used in cancer diagnostics, constitutional testing, as well as prenatal testing. In 2012, RNAScope was invented as the next generation RNA in situ hybridization technology. This was a true advancement in unlocking the detection of single RNA molecules within single cells with low abundance RNA detection, something that previous technologies was unable to do. As a result, there has been a wide range of RNAscope adoption in laboratories for plenty of meaningful application. Now, let's take an overview of how RNAscope has evolved as most advanced next generation ISH technology and how different it is from the conventional in situ hybridization in terms of sensitivity and specificity. In traditional in situ hybridization, single standard probes are labeled with a molecule called deoxygenin, which is shown here in aqua color. This one. These probes are subsequently detected by anti-deoxygenin antibodies and then in turn secondary antibodies which are conjugated to horse redox peroxidase enzyme or HRP. HRP can then act on the substrate DAB. As many of you are familiar, DAB will then produce a brown chromogen at the target site. This technique works quite well for some targets, particularly for those which are expressed at high levels. However, this technique also has several disadvantages. The main disadvantage of this is because it is a direct labeling technique, and it produces only weak signals in absence of high RNA expression. This doesn't allow for significant amplification of the signal. You can only load so much of deoxygenin into these probes, which limits the amplification as well as the ability of this technique to detect low expression targets. Another disadvantage of this traditional technique is that it has the potential to produce relatively high levels of background staining due to the non-specific binding of the in situ hybridization probes to sequences with only partial homology to the actual probes. This can make interpretation and distinguishing the true signal from the background very challenging. Now that we have talked about the traditional in situ hybridization, let us learn how this assay has progressed to the next generation RNAscope in situ hybridization. RNAscope is a new generation of RNA in situ hybridization with better sensitivity and specificity. The key to the innovation is in the probe design, which we refer to as the double Z design. This is a Z probe, and each of these Z target probe contains 3 elements. The lower region or the bottom part, which is complementary to the target RNA, it is only in double-digit base pairs. So it is very small and enables us to have a high degree of sensitivity. A linker sequence that links the lower region to the upper region and the top part of the Z, which serves as a binding site for the pre-amplifier sequence. So in the first step, two adjacent Z probes hybridize to the target RNA in tandem in order for signal amplification to occur. A pre-amplification base then binds to the site formed on the top of each double Z pair. The pre-amplifier sequence is nothing but a DNA sequence. Next, amplifiers, which are also DNA sequence, then attach to the binding site on each pre-amplification base. Finally, label probes are in the form of chromogenic enzymes or fluorophore is applied to allow visualization of amplified target probe hybridization. To add on, the probe design is designed to typically have 20 ZZ pairs per target, per gene, basically. And with the 20 pairs, you will get a massive signal amplification with an exquisite sensitivity all the way down to a single RNA molecule, which allows us to have the robust sensitivity. Now, if you remember, in traditional in situ hybridization methodology, which I have just described in the earlier slide, probes tend to be quite large or long. And because of that, they are less susceptible to mismatches in DNA, which contributes to a meaningful amount of background signal. With RNAscope, it is always necessary for two of the Z probes to hybridize adjacent to each other in order for the pre-amplifier base to bind. Because of how small the binding site is to the target sequence, it is more susceptible to mismatches. And if, say, only one Z probe were to bind, pre-amplifications cannot occur. This prevents amplification of non-specific signals, contributing to the high specificity of this next generation in situ hybridization technology. Here I have listed a few of the ACD probe which are clinically significant. So I have categorized them, as you can see, as per the clinical applications. And there are several of them for different kind of pathological situation. So this is especially for the clinical probe menu. But for the research, we have unlimited possibilities. In our catalog, we have over 55,000 probes across 400 different species. And all these probes are available in our online catalog, which could be available at www.acdbio.com. But we are not restricted to that. If you are working on any novel pathogen, novel gene sequence, we can generate custom probes for any gene within any species within 2 weeks. And therefore, we address that RNAscope is universal. It can be applied for any gene in any species in any tissues. And on top of that, you would be able to do a multi-omics analysis where you could be able to detect RNA protein together on the same sample. That means you could be able to stain your RNA markers along with your protein markers, which are using the antibodies on the same sample. Now, let me walk you through some unique applications of RNAscope from all the published articles. Here is a flagship application of RNAscope for oral and cervical cancers. So RNAscope HPV probes are being used to detect the direct involvement of HPV for this kind of cancers. The current diagnostic approach uses P16 immunohistochemistry, which is a surrogate marker for detecting the HPV involvement in oral and cervical cancers. Because until and unless the HPV, which is the virus, expresses the E6, E7 mRNA, it is not oncogenic. So mere presence of P16 positive samples doesn't tell us that HPV is positive for those kind of cancers. We have probes to target the E6, E7 mRNA for each of the HPV subtypes, We also have cocktails of probe which cover multiple high-risk and low-risk subtypes. Here is a classical staining examples of our high-risk cocktail, which contains 18 different high-risk HPV subtypes. And from the publications, this is a summary of comparison of all the tissue-based diagnostic approach for oral and cervical cancers and comparing RNAscope with P16IHC or DNA-ish. This is the most sensitive and specific method, which is RNAscope, and this is the only method which provides you a direct evidence of HPV activity through the E6, E7 mRNA detection. Hence, most of the clinical experts consider RNAscope-ish as gold standard for determination of HPV status in oral and cervical cancers. Now, here is a classical example of expression of microRNA 205 and associated tumor target genes in head and neck cancer tissue. Downregulation of tumor suppressor TP53, which is shown here in green color, TP53, resulted in upregulation of microRNA 205, which is the marker here, and that in turn downregulates P10 expression, which is here. And pan-cytokeratin stained the whole tumor region in the tissue, which is right here. Okay. Next, this is a classical example of using RNAscope probes for B-cell lymphomas by detecting the immunoglobulin kappa and lambda light chains. So on the top panel, you can see the IHC. So this is a case which is lambda restricted, and you can see the background of kappa in using the antibodies. Whereas the same sample, when it was detected by RNAscope kappa and lambda probes, you can see how clean is the background. So it was very easy for the clinicians or the pathologists to score this sample as the lambda restricted B-cell lymphoma. Similarly, in this slide, you can see the left panel is the RNA IHC using the antibodies, and the right panel is RNAscope. So this is a kappa-restricted lymphoma, and you can see how clean is the background for RNAscope compared to IHC, which has a very high background, and it was difficult to say whether it's polyclonal or it's a kappa-restricted lymphoma. Now I'm moving on to another example of using MDM2 RNAscope probes for detection of sarcoma. MDM2 is an oncogene which controls the P53 activity, and the typical cancers which are associated with MDM2 amplification is liposarcoma, atypical lipomatous tumors. RNAscope in situ hybridization for MDM2 is equivalent to superior performance when compared to DNA fish for identification of MDM2 amplification in this kind of cancers. Next, I'll show you a great example of albumin RNAscope that addresses the high background of immunohistochemistry. Albumin is a major serum protein, and consequently, it is present at low levels in almost all vascularized tissues, contributing to high background with albumin immunohistochemistry. RNAscope gets around this issue with multiple groups publishing on the utility of albumin RNA in situ hybridization in the differentiation of primary liver tumors from intrahepatic cholangiocarcinoma and metastatic adenocarcinomas. Rounding out some of the more widely utilized infectious disease probes, our menu also includes Epstein-Barr virus and cytomegalovirus probes. Publications have demonstrated increased sensitivity in the detection of these viruses when compared to traditional immunohistochemistry, which is especially useful in the diagnosis of associated gastric carcinoma and various B-cell lymphomas, which I'll give you an example in the next slide. While several groups reported on their ability to detect CMV, others were unable to replicate this finding, raising questions about the assays used and their sensitivities for CMV detection. The ongoing controversy of CMV detection in gliomas has significant clinical implications, since suppression of CMV replication with antiviral therapy could potentially modify the prognosis of these cancers. If CMV plays a role in glioblastoma, future clinical trials could potentially support changes to its standard of care. In addition to antiviral therapy, the presence of CMV in glioblastoma may suggest that immunotherapies such as anti-CMV vaccines might be of value. Here is a classical example of RNAscope detection of MYC-N in neuroblastoma. So as you can see, RNAscope was successfully able to detect the different grades. With the lowest grade, you can still see the RNAscope punctures or the brown colored dots. And the authors claim that MYC-N RNA could be a better biomarker than MYC-N amplification for predicting poor prognosis of neuroblastoma patients. This is a wonderful example of validating the heterogeneity of high-risk neuroblastoma using the RNAscope probes. And as you can see, the MYC-N RNA with the different cell type-specific markers are being visualized on the same samples using the fluorescent RNA in situ hybridization assays. And this is also a wonderful example of using RNAscope technology to validate the transcriptomics data. Moving from this application, I will showcase you the ability of RNAscope probes to detect the protein isoforms or exon junctions. This is an example of detecting the EGFR8 variant in glioblastoma. So in this slide, we show two glioblastoma cases, this left one and the right one, which are stained with the probes specific for wild type. So this is the wild type probe. As you can see, it says it targets the E1, exon 1 and exon 2 junction, and the exon 1 and exon 8 mutant probe. So this is the EGFR8 mutant probe. With the wild type probe, we can see staining in both the cases. So here and here, you can see staining in both the cases. However, with the mutant probe, we can only see signal for the EGFR positive glioblastoma case, but not for the EGFR negative glioblastoma case, taken together, we can specifically identify the EGFR8 variant in the FFP glioblastoma samples. Here is a classical example of RNAscope detecting the point mutations, where we are detecting the IDH1 arginine to histidine mutation at 132 position. And you can see that the point mutation probes were being validated in cell blocks. And after that, we move to the different tissues. So in our duplex assay, colorimetric duplex assay, which can detect two RNAs, you can see the mutant probe as well as the wild type probe in two different colors, the green and the red colors. And you can see their expression across the different astrovitoma, astrocytoma, and glioma tissues. So RNAscope can also be used to detect point mutation. And here is a list of the probes which are available for point mutation in our catalog. This is a unique application of RNAscope for ovarian cancer. And you can see here the lymphoid and myeloid immune infiltration with cytokine detection. So you can see here cytokines like interferon gamma can only be detected using the ELISA technique, because these cytokines, when they are translated to proteins, they secrete out of the cell. But we can detect them using RNAscope technology because we are detecting the mRNA, which is before the protein expression. And you can actually see, using the CD8 and interferon gamma combination, cytotoxic T lymphocyte in the tumor microenvironment, which is actively releasing the interferon gamma. So this is a very unique application of RNAscope to detect the cytokines and to check the immune infiltration in your tumor microenvironment. Now we are moving into a very interesting application of this technology, where you can stain RNA and protein on the same tissue section. Same thing like here, you can see this is a lung cancer tissue, where you can stain the PD-L1 RNA mRNA, and the CD45 protein. So the RNA is depicted in the red color and the protein is depicted in the blue color. So you can see the nice distribution of the tumor and stroma using the RNA and protein code staining. Here is also an example of the same application, RNA protein co-detection, but here we are detecting a microRNA and antibody, which is microRNA 223 and CD3 antibody. So you can see clearly T cell infiltration within cervical tumor can be visualized using the CD3 immunohistochemistry in combination with the myeloid-specific microRNA 223 using the microRNAscope probe. Now, let me walk you through our comprehensive suite of spatial biology solutions that enable visualization of virtually any RNA species alongside the proteins. At the core of our technology portfolio is RNAscope, designed for detecting mRNA, long non-coding RNA, basically any RNA targets over 300 bases. Complementing this, we have BaseScope, which specializes in shorter targets between 50 to 300 bases, making it ideal for detecting splice variants like the EGFR8 variant and point mutations. For the smallest RNA species, like microRNA, silencing RNA, and antisense oligo, we offer microRNAscope technology. What makes our platform truly powerful is the ability to combine any of these RNA detection methods with protein visualization through our RNA protein co-detection assay. This integration creates a true spatial multiomics approach, providing unprecedented insights into cellular mechanisms. Our technology has become essential tool across multiple research fields with particularly strong adoption in cancer science, neuroscience, inflammation and immunology, developmental biology, cell and gene therapy, infectious disease. Throughout today's presentation, I have shared specific examples of how researchers and clinicians in these fields are leveraging our technology to make groundbreaking discoveries and improving the clinical diagnostics. Now, I will wrap up by sharing some impressive numbers that highlights RNAscope position as the gold standard for in situ hybridization as well as RNA protein co-detection applications. Our global team includes over 250 RNAscope and multiomics experts, supported by 60 plus PhD scientists who specializes in probe design, field applications, and technical expert. Our extensive catalog now features more than 55,000 unique RNA probes covering over 400 species across 50 different countries. This breadth of coverage demonstrates our commitment to serving diverse research needs worldwide. The impact of RNAscope technology is evident in our publication record, with more than 11,000 peer-reviewed papers published since its inception in 2011. Till date, we have over 11,000 peer-reviewed publications. Perhaps more significantly, RNAscope has been utilized to stain over 250,000 slides for clinical diagnostics, underscoring its reliability in critical diagnostic application. These matrices reflect not just our established presence in spatial biology, but also our ongoing commitment to advancing the technology while maintaining the highest standards of sensitivity and specificity that clinicians and researchers have come to expect from us. With that, I have come to the end of this presentation, but before I end, on a sweeter note, I would like to highlight everyone the services which are being provided by biomarketing services in Malaysia. So you can have lab services for ACD or to adopt RNAscope technology, and you can send your samples to BMS lab. And to add on to that, here are a list of projects which we have successfully delivered in Malaysia. Some of them are completed, some are ongoing. And with that, I would like to thank you everyone for your patience, listening to me, and giving me the opportunity to present this unique technology and its applications to the group. Thank you.
Yeah, thank you, Dr. Amrita, for the very interesting talk. First, before we progress to the question, I want to stress that I'm not being paid by the company or have any interest in the company. Just came across this presentation and I thought this was a very interesting technology. Because every time we deal with RNA specimens, the RA have to be in OT, they have to take the sample fresh, then they have to rush to fix it, and it's like such a mess. But if I'm not mistaken, this can be done on FFPE, formalin fixed paraffin embedded tissue, right?
Absolutely, doctor.
Yeah,
This
yeah.
technology
So
was invented to
yeah.
address that RNA loss during extraction.
Yeah, so this is actually quite fantastic. You don't need to come and harass us to snap freeze the tissue, and
I
that
agree.
urgency is not there. So I thought I really had to share it with everybody in the hospital. But in terms of utility in reporting, I don't think any hospitals in Malaysia has put it as part of their reporting. Is it a commonly used technique in Malaysia? I actually don't think I know of any hospitals in KKM or private that's using this.
That there is a reason for that, doctor. So in America and Europe and parts of Japan and Australia, we are IBD, but our probes are not yet IBD in Asia. So what
Ah,
happens is in other countries like Singapore, like India, Thailand, they are using our RUO probes, the one which I listed as clinical probes, and they are using it as a lab developed test to report patient data. So they
ah,
have taken a few of the patient samples, let's say 20 to 30, and they have validated the application, and then they have submitted the document to their authority so that that can be granted, that test can be granted as a CDX or clinical diagnostics in that particular lab. So in Malaysia, we yet do not have any, but our assays are being run, as I showed you, our assays are being run by IMH, NIH, and they are validating the assay probably to bring it through the clinics.
okay. Yeah, I think we can go to the question. There is one in the. I'll do the chat box one first because that came in earlier. It says, "Hi, thanks for this talk. Is this provided as service or can be done?" No, it's not for service. What are the specific instruments required? Is there a specific instrument or can we run it on our Ventana benchmark or piggyback on the cheat
Good
and
question, doctor. So this is
piggyback?
provided as a service by BMS,
I see.
basically. Yeah.
But the
So
equipment wise,
equipment-wise, it is compatible with Leica Bond 3 and Ventana Discovery Ultra.
it's kind of big machine, huh? Okay, and then the next, what did he say? What is the highest multiplex capacity maximum number in single samples?
Okay, so if you are talking about FFP samples, we can go up to 12 RNA detection, but that is using the fluorescent assay. But this 12 RNA detection, you do not need any special microscope. You can do it with your standard fluorescent or confocal microscope. And for fresh samples, which is fresh frozen or fixed frozen tissues, we can go up to 48 different RNA detection within the same tissue. And
Oh,
again, you do not need any special microscope for that.
oh, okay. And then the next part of the question also is, can this be used for mRNA specific for isoforms?
Yes, definitely. So that's our base scope example, which I have shown you in the EGFR glioblastoma case. We
Mm,
can
mm,
detect splice variants or mRNA variants or even stem cell variants. So basically, any exon junction can be targeted using these probes.
Okay. And then for the Q&A, there is one question here. It sounds like for IVD, is this method recommended by international guidelines? Is there anything in the international
Yes.
guidelines about
Yes.
this?
So we have NCCN guidelines and CAP guidelines, especially for our HPV detection. So NCCN guidelines, which is 2023, it clearly says that PCR and in situ hybridization are the two recommended assay. which will provide sensitivity and specificity for HPV involvement in oropharyngeal cancers. And the CAP guidelines, which is from 2018, they said that in the coming days, HPV RNAscope is going to be the chosen assay for HPV involvement in oropharyngeal and squamous cell carcinomas.
Oh,
So these are
that's
the two
nice.
guidelines. If someone wants, I can share that. I have slides on that.
Okay, okay. So the participants can please take note of that, or you can come and ask me. I'm Dr. Man of Pathology. Thank you again, Amrita, and also thank you to Dr. Aiman and the ENT speaker. Oh, one thing about the ENT speaker, the previous one, the mention of can polyps transform into malignancy. Yeah, I totally agree with the speaker. Allergic nasal polyps don't transform. They're usually benign. But there are many tumors that can masquerade as a mass in the nose. And because my field is pathology, I just recently had an inverted papilloma that has turned malignant and a meningioma of the brain that has extruded into the nose and masqueraded as a nasal polyp. So we really depend on the ENT expertise when to biopsy, when to remove. But if you have anything that's blocking your nose, I think please go and see them as soon as possible so that we can get the biopsy sample. Yeah. Okay. And thanks again, Dr. Amrita and everybody. Yep. I
Thank you,
think
everyone. If you want to know more about the services of RNAscope being done in Malaysia, there is a QR code on the slide. You can just scan it.
that's wonderful. That's wonderful. Okay. All
Thank
right.
you so much, Prof. Man. Thanks,
Yep.
everyone.
Okay. Thank you, everybody. I think we can call it a wrap. Yep. Thank you again for all the speakers and have a good day. And for our Muslim friends, selamat berpuasa. Okay. Bye-bye.